Hello students, today we will learn about microbial production of vitamins. Vitamins. Vitamins are organic compounds that perform specific biological functions for normal maintenance and optimal growth of an organism. These vitamins cannot be synthesized by the higher organisms, including man, and therefore they have to be supplied in small amount in the diet. Microorganisms are capable of synthesizing the vitamins. In fact, the bacteria in the gut of humans can produce some of the vitamins which, if appropriately absorbed, can partially meet the body's requirements. It is an accepted fact that after administration of strong antibiotics to humans, additional consumption of vitamins is recommended. Microorganisms can be successfully used for the commercial production of many of the vitamins. Example: thiamine, riboflavin, pyridoxine, folic acid, pantothenic acid, biotin, vitamin B12, ascorbic acid, beta carotene, that is provitamin A, ergosterol, that is provitamin D. However, from economic point of view, it is feasible to produce vitamin B12, riboflavin, ascorbic acid, beta carotene by microorganism. For now, let's know about vitamin B12. From starting, we will discuss about vitamin B12. The disease pernicious anemia, characterized by low levels of hemoglobin, decreased number of erythrocytes, and neurological manifestations, has been known for several decades. It was in 1926 some workers reported the liver extracts could cure pernicious anemia. The active principle was later identified as vitamin B12, a water-soluble beta-complex vitamin. Now let's know about the occurrence. Vitamin B12 is present in animal tissue at a very low concentration. It occurs mostly in the coenzyme forms, that is methyl cobalamin, and cobalamin. Isolation of vitamin B12 from animal tissues is very expensive and tedious. Now, let's know about the chemistry. Vitamin B12, that is cyanocobalamin, is a water-soluble vitamin with complex structure. The empirical formula of cyanocobalamin is C63 H90 N14 O14 PCO. The structure of vitamin B12 consists of a corine ring structure with a central cobalt atom. The corine ring is almost similar to the tetrapyrrole ring structure found in other porphyrin ring compounds. Example: heme with Fe iron and chlorophyll with Mg. The corine ring has 4 pyrrole units. Cobalt present at the center of the corine ring is bonded to the 4 pyrrole nitrogens. Cobalt also binds to dimethylbenzimidazole and aminoisopropanol. Thus, cobalt atom present in vitamin B12 is in a coordination state of 6. Now let's know about biosynthesis. Vitamin B12 is exclusively synthesized in nature by microorganisms, and the biosynthesis of B12 is comparable with that of chlorophyll and hemoglobin. Many of the reactions in the synthesis of vitamin B12 are not yet fully understood. Now let's know about commercial production of vitamin B12. Vitamin B12 is commercially produced by fermentation. It was first obtained as a byproduct of streptomyces fermentation in the production of certain antibiotics, streptomycin, chloramphenicol, or neomycin, but the yield was very low later high yielding strains were developed. And at present, vitamin B12 is entirely produced by fermentation. It is estimated that the world's annual production of vitamin B12 is around 15,000 kg. High concentrations of vitamin B12 are detected in sewage sludge solids. This is produced by microorganisms. Recovery of vitamin B12 from sewage sludge was carried out in some parts of the United States. Unlike most other vitamins, the chemical synthesis of vitamin B12 is not practicable since about 20 complicated reaction steps need to be carried out. Fermentation of vitamin B12 is the only choice. Microorganisms and yields of vitamin B12. Let's know about microorganisms and yield of vitamin B12. Several microorganisms can be employed for the production of vitamin B12 with varying yields. Glucose is the most commonly used carbon sources. The most commonly used microorganisms are Propionibacterium, Freudenrichi, Pseudomonas denitrificans, Bacillus megaterium, and Streptomyces olivacea. Genetically engineered strains for vitamin B12 production. Let's know about genetically engineered strains for vitamin B12 production. By employing modern techniques of genetic engineering, vitamin B12 can be enhanced. A protoplast fusion technique between Protaminobacter rubber and Rhodopseudomonas spheroids resulted in a hybrid strain called Rhodopseudomonas protamicus. The new strain can produce as high as 135 mg per liter of vitamin B12 utilizing carbon sources. Now let's know about production of vitamin B12 using Propionibacterium species. Propionibacterium Freudenrichi and P. Sermani and their mutant strains are commonly used for vitamin B12 production. The process is carried out by adding cobalt in two phases. Now let's know about anaerobic phase. This is a preliminary phase that may take 24 days, 2 to 4 days. In the anaerobic phase, 5'-deoxyadenosylcobalamin is predominantly produced. Aerobic phase. Let's know about aerobic phase. In this phase, 5,6-dimethyl is produced from riboflavin, which gets incorporated to finally form coenzyme of vitamin B12, namely 5'-deoxyadenosylcobalamin. In recent years, some fermentation technologists have successfully clubbed both an anaerobic and aerobic phases to carry out the operation continuously in two reaction tanks. The bulk production of vitamin B12 is mostly done by submerged bacterial fermentation with beet molasses medium supplemented with cobalt chloride. The specific details of the process are kept as a guarded secret by the companies. Now let's know about the recovery of vitamin B12. The cobalamins produced by fermentation are mostly bound to the cells. They can be solubilized by heat treatment at 80 to 120 degrees centigrade for about 30 minutes at pH 6.5 to 8.5. The solids and mycelium are filtered or centrifuged and fermentation broth collected. The cobalamins can be converted to more stable cyanocobalamins. This vitamin B12 is around 80% purity and can be directly used as a feed additives. However, for medical use as a feed additive, However, for the medical use, but, for medical use, vitamin B12 should be further purified. That means 95 to 98% purity should be there. And now let's know about production of vitamin B12 using Pseudomonas species. Pseudomonas denitrificans Pseudomonas denitrificans is also used for large-scale production of vitamin B12 in a cost-effective manner. Starting with a low yield 2 decades ago, several improvements have been made in the strains of denitrificans for a tremendous improvement in the yield. Addition of cobalt and 5,6-dimethyl benzimidazole to the medium is essential. The yield of vitamin B12 increases when the medium is supplemented with B10. Carbon sources for vitamin B12 production. Let's know about carbon sources for vitamin B12 production. Glucose is the most commonly used carbon source for large-scale manufacture of vitamin B12. Other carbon sources like alcohols, methanol, ethanol, isopropanol, and hydrocarbons, that is alkenes, decane, hexadecane with varying yield can also be used. A yield of 42 mg per liter of vitamin B12 was reported using methanol as the carbon source by the microorganism Sarcina barkeri in feed batch culture system. Now let's know about riboflavin. Riboflavin, it's a vitamin B12 is a water soluble vitamin essential for growth and reproduction in man and animals. Deficiency of riboflavin in rats causes growth retardation, dermatitis, and eye lesions. In humans, vitamin B2 deficiency results in chelosis. That means fissures at the corner of mouth, glossitis, that is purple tongue, and dermatitis. Riboflavin exerts its biochemical functions through the coenzyme, namely flavin adenine dinucleotide and flavin mononucleotides. Now let's know about the occurrence. Riboflavin occurs in milk and milk products, meat, eggs, liver, and kidney. While in milk and eggs it is present in free form, in other foods it is found in the form of riboproteins, flavoproteins, that is coenzymes of riboflavin bound to proteins. Now let's know about chemistry. Riboflavin contains 6,7-dimethyl isoalloxazine attached to d-ribitol by a nitrogen atom. The isoalloxazine ring participate in the oxidation reduction reactions brought out by the coenzymes FAD and FMN. Now let's know about biosynthesis. The biosynthetic pathway of riboflavin elucidated for the microorganism Asbia gossypii and Eremothecium asbi. The overproduction of riboflavin in these organisms takes place mainly due to the constitutive nature of the riboflavin synthesizing enzymes. Iron, which inhibits the production of vitamin B12 in the Clostridia and yeast, has no effect on A. gossypii and E. asbis. Now let's know about commercial production of riboflavin. There are three processes employed for the large-scale production of riboflavin. The worldwide requirement of riboflavin estimated to be around 2,500 tons per year. Now let's know about biotransformation. About 50% of the world's requirement of riboflavin is produced by biotransformation, followed by chemical synthesis. For this purpose, glucose is first converted to D-ribose by mutant strains of Bacillus pumilus. The D-ribose so produced is converted to riboflavin by chemical reactions. Second is chemical synthesis. Approximately 20% of the worldwide riboflavin is produced by direct chemical synthesis. Third is fermentation. At least one-third of world's riboflavin requirements are met by direct fermentation process. Microorganisms and yields of riboflavin. Let's know about microorganisms and its Yields of riboflavin. Several microorganisms like bacteria, yeast, and fungi can be employed for the production of riboflavin. In the acetone butanol fermentation, employing the organisms Clostridium acetobutylicum and Clostridium butylicum, riboflavin is formed as a byproduct. Commercial production of riboflavin is predominantly carried out by direct fermentation using the ascomycetes. The two plant pathogens, namely Asbia gossypi and Eremothecium asbi, are most commonly employed due to high yield. Among these two organisms, A. gossypi is preferred as it is more stable with a high producing capacity of riboflavin. Now let's know about genetically engineered strains for riboflavin production. High yielding strains of gossypi have been developed by genetic manipulation, and such strains can yield as high as 15 gram per liter riboflavin. Now let's know about the production process of riboflavin. Industrial production of riboflavin is mostly carried out with the organism Asbia gossypi by using simple sugars such as glucose and corn step liquor. Glucose can be replaced by sucrose or maltose for the supply of carbon sources. In recent years, lipids such as corn oil, when added to the medium for energy purpose, have a profound influence on riboflavin production. Further supplementation of the medium with yeast extract, peptones, glycine, inositol, purines also increase the yield of riboflavin. It is essential to carefully sterilize the medium for good yield of riboflavin. The initial pH of the culture medium is adjusted to around 6 to 7.5. The fermentation is conducted at temperature 26 to 28 degrees centigrade with an aeration rate of 0.3 ppm. The process is carried out for about 5 to 7 days by submerged aeration fermentation. Riboflavin fermentation by Eremothecium asbi is comparable to that described until now for Asbia gossypi. Candida species can also produce riboflavin, but this fermentation process is extremely sensitive to the presence of iron. Consequently, iron or steel equipments cannot be used. Such equipment have to be lined with plastic material. Fermentation through phases. Let's know about fermentation through phases. Some studies have been carried out to understand the process of fermentation of riboflavin, particularly by ascomycetes. It is now accepted that the fermentation occurs through three phases: phase 1, phase 2, phase 3. Phase 1. This phase is characterized by rapid growth of the organism utilizing glucose. As pyruvic acid accumulates, pH becomes acidic. The growth of the organism stops as glucose gets exhausted. In phase 1, there is no production of riboflavin. Now, phase 2. Sporulation occurs in this phase and pyruvate concentration decreases. Simultaneously, there is an accumulation of ammonia due to enhanced deaminase activity, which makes the medium alkaline. Phase 2 is characterized by maximal production of riboflavin, but this is mostly in the form of FAD and a small portion of it as FMN. Phase 3. In this last phase, cells get disrupted by the process of autolysis. This allows release of FAD, FMN and free riboflavin into medium. Now let's know about recovery. Riboflavin is found in fermentation broth and in a bound form to the cells. The latter can be released by heat treatment, that is 120 degree centigrade, for about 1 hour. The cells can be discarded after filtration or centrifugation. The filtrate can be further purified and dried as per the requirements. Now let's know about the other carbon sources for riboflavin production. Besides sugars, other carbon sources have also been used for riboflavin production. A pure grade of riboflavin can be prepared by using Saccharomyces species, utilizing acetate as sole carbon source. Methanol, utilizing organism Hansenula polymorpha, was found to produce riboflavin. The other carbon sources used with limited success for riboflavin production are aliphatic hydrocarbons. Now let's know about beta carotene. Beta carotene is a provitamin A. When ingested, it gets converted to vitamin A in the intestine. Vitamin A is a fat soluble vitamin required for vision, proper growth, and reproduction. The deficiency of vitamin A causes night blindness, changes in the skin and mucosal membranes. Now let's know about occurrence and chemistry. Beta carotene is found in many animal and plant tissues. However, it originates exclusively from plants or microorganisms. Yellow and dark green vegetables and fruits are rich in beta carotene. Example: carrots, spinach, amaranthus, mango, papaya. Carotenoids are isoprene derivatives. Chemically, they are tetraterpenoids with 8 isoprene residues. There are around 400 naturally occurring carotenoids. The most important carotenoids are beta carotene, alpha carotene, delta carotene, and lycopene and zeaxanthin. Xanthin. Zeaxanthin. Carotenoids are mainly used as coloring agents. Example: beta carotene, lycopene, xanthophylls. Several food like cheese, meat, egg products can be made attractive by coloration. It may be noted that the demand for beta carotene as the provitamin A is comparatively less. Now let's know about biosynthesis. The pathway for the biosynthesis of beta carotene and some other important carotenoids we will discuss in plants and fungi. Let's know about commercial production of beta carotene. Beta carotene can be produced by microbial fermentation. However, for economic reasons, direct chemical synthesis of vitamin A is preferred rather than using its provitamin beta carotene. Now, let's know about the microorganisms. The organisms black, Blackesia, Trispora, Trispora, that is, and Phycomyces, Blackes linus, and Coanephora cucurbitarium are most frequently used for the production of beta carotene. Among these, Blackesia trispora is preferred due to high yield. Some important carotenoids, the organisms, and the production yield are now we will discuss. First organism, beta carotene, that is Blackesia. Production yield is 3.0. Lycopene, that is Blackesia, Blackesia trispora, it's 0.4. Its production, its yield, and xanthin, that is Flavobacterium species, that is 0.4. Streptomyces Crestomyceticus, it's 0.5. Its production yield is 0.5. Now let's know about the production process of beta carotene. The industrial production of beta carotene is mostly carried out by Blackesia trispora. The fermentation medium contains cornstarch, soybean meal, beta ionon, antioxidants, etc. Addition of antioxidants improves the stability of beta carotene within the cells. The fermentation is carried out by submerged process. The fermentation is usually started by mixing the cultures of both sexual forms plus and minus strains of beta trispora. The yield of beta carotene is significantly higher with mixed cultures compared to plus or minus strains. This is due to the fact that beta carotene production predominantly occurs during the process of zygospore formation. It may be stated here that the use of mixed strains does not improve the yield for other microorganisms. And let's know about factors affecting production. Trisporic acid, which can act as a microbial sexual hormone, improves production yield of beta carotene. Beta ionons enhance beta carotene synthesis by increasing the activity of enzymes and not by their direct incorporation into beta carotene. When the fermentation medium is supplemented with purified kerosene, beta carotene production is almost doubled. Kerosene increases the solubility of hydrophobic substrates. Now let's know about recovery. The mycelium rich in beta carotene can be directly used as a feed additive. For purification, mycelium is removed, subjected to dehydration and extracted in methylene chloride. This product is of 70 to 85% purity, which can be further purified as per the requirement. Now let's know about gibberellin, that is plant growth stimulating hormone. Gibberellins are plant hormones that stimulate plant growth. They promote growth by cell enlargement and cell division. The observable effects of gibberellins include stimulus to seed germination, flowering and lengthening of stems. Now let's know about microbial production of gibberellin. So far, only one microorganism, that is the fungus, namely Gibberella fujikuroi, has been found to produce gibberellins. This is actually pathogenic fungus of rice seedlings. Gibberellin production can be carried out by using a glucose salt medium at pH 7.5 and temperature 25 degrees centigrade for 2 to 3 days. The fermentation process is conducted in aerated submerged process. After the growth of the fungus is maximum, the production of gibberellins commences. Finally finished. Any questions regarding this? You can ask me in the comment box and subscribe my channel to know more updates. Thank you.